ASRB Net 2024 preparation model paper 2 related to history of plant pathology. So as I said earlier, at least we do expect at least 5 to 10 questions from this history of plant pathology. So studying history related to semester related exams or studying history related to other kinds of exams are different when compared to net exam. So they will ask questions like for I'm just telling all the discoveries and their contributions towards the pathology. You guys remember they will ask any kind of questions like what is the book written by the particular scientist, or maybe who discovered this particular thing, or maybe who actually named this thing. Such kind of things we do expect. So be very careful while going with the history, because if you have not yet not well versed about the historical events that happened, so this is the time, and we have placed a very good amount of history, like we have collected all the information from the various subjects like mycology, bacteriology, virology, epidemiology, applied plant pathology, biocontrol agents, and all the things that we have collected and placed here. The best points are the most important points, not most important points, I would say the almost all kind of contributions that are related to the particular scientist and all those things that we have placed here itself. It will be definitely helpful. Now we'll start with this. I'm Anish Shankar Reddy, PhD plant pathology. We'll starts with mycology. The first one is Theophrastus. Generally, Theophrastus is considered as the father of botany. He wrote books like Inquiry into the Plants, the nature of plants, and the reasons of vegetable growth. Actually, the nature of plants are the inquiry into the plants almost more or less similar, but the reasons of vegetable growth. So in the questions will be like, who is father of botany? Name the book that was written by Theophrastus, or else they will ask like nature of plants was written by, reasons of vegetable growth was written by, such kind of things. He told that punishment of plant diseases are the general the punishment or wrath of gods. And this question was I have in the previous books I have read this question like. Who told that plant diseases are the punishment or wrath of God? Wrath means punishment only. Wrath or punishment of God, that was given by Theophrastus only. He is Theophrastus. So two questions we do expect from this. Father of botany, or maybe inquiry into the plants was written by who, and who told that plant diseases are wrath or punishment of God? Such kind of questions we do expect. The next one is Democritus at the time. He recommended that spraying of olive oil for the management of plant blights. And Homer at 1000 BC, he also mentioned the therapeutic properties of sulfur in managing of plant diseases. So they will ask who mentioned that therapeutic properties of plant diseases for the first time, Homer. And who mentioned that, or who gave that sprinkling of olive oil can control the blights? So Democritus 470 BC, something like that they will ask. Okay. And the next one is Robigolia. This is a very important one. Generally, what will be the probability questions? See, if I start telling you the history, it will be a very big thing, right? So I'm not dwelling in deep into the history, because history is a very important a very interesting aspect for me, so that I have dwelled very deep into the history. But since now we have very time, I'm not telling all the facts because that ran out of time. So that I will tell you the what is the questions of the exam importance. Okay. Robigolia, generally that was initiated by a king called King Numa Pompilius. That was actually initiated by a king, King Numa Pompilius. In nowadays we are celebrating on April 25th of every year. So Romans generally used to celebrate this to ward off rust. So to protect their crop from rust diseases, they have created a god called Robigus. So they used to celebrate a festival called Robigolia. At the time they used to sacrifice like cows, red oxes, dogs, and all the kind of things they used to sacrifice. So that was the only probability question we can expect is on which date, or maybe what is the date that used to celebrate the festival called Robigolia. So April 25th, the god name is Robigus. Okay. In the picture we can see the kind of animals the people are offering to Robigus to calm down or to not infect their crops. Such kind of belief was there in that time. So it was initiated by King Numa Pompilius around 700 BC, I think so. The next one is Albertus Magnus. He's the first man. He's not actually a scientist. He's actually a church priest. He was the first person who recognized the world's first plant pathogen in the world, that is mistletoe. Now we can call it as a Loranthus, right? So the first plant pathogen in the world that was discovered by Albertus Magnus, that is called mistletoe. We can also call it as Loranthus, also called as Dendropthy. and generally infect common leafy vegetables. European mistletoe is called viscum, Northern American mistletoe is called phonodendron, dwarf mistletoe is called archithobium. And in Nicander, one more person, he called the mushrooms are the evil fermenters of the earth. They will put any sentence and they will ask who discovered it or who formulated it, some kind of questions we do expect. Okay, we must be very careful on it, right? Next one, Thaulier, a first man. Actually, this ergotism is a very old disease, and at the time people used to believe that they have no idea. People used to believe that it was a god curse. That is the only thing what they know. And later it was identified by the ingestion of some kind of eating infected kind of grains. That's why it is called a holy fire. If you see the the symptoms of the infected persons, we can see some kind of firing kind of symptom. We can observe here the hands look like fire, right? That's what's called as holy fire. Ergotism, holy fire or Saint Anthony's fire, various kinds of things were. That was first discovered by a French physician, Thaulier. He's actually a doctor actually. Ergot in general, ergot or argot is a French word which means spur. It is generally ergot is mainly due to caused by claviceps purpurea. People at the time used to believe, as I told you, that is a devil's curse, God's curse. We can also call it as devil's curse, who are doing the wrong kind of things or wrong sins. God gives such kind of disease, such kind of things. At the time, people used to believe. It can also called as fire or holy fire. As I told you, the infected persons, you can take limbs, especially hands and legs. It look like fire. That's why it's called as holy fire or Saint Anthony's fire. And ergot alkaloid, ergot producing an alkaloid called LSD, lysergic acid diethylamide. It is generally called as drug of the century because this LSD was used to cure the mental disorder persons. So it is one of the very good drug that can be used to cure this mentally disordered persons. And this is about some details regarding to ergot. We expect like ergotism is mainly due to which fungi? Claviceps purpurea, right? So otherwise called as holy fire, Saint Anthony's fire. Ergot producing alkaloid, LSD, lysergic acid diethylamide. Okay, it is called as a drug of the century by Bow in 1970 because as I told you that it is used to cure the mentally disordered persons. And 1601, Clusius, he actually drawed mushrooms, watercolor drawing of mushrooms, and he wrote a book called Rarorium Plantarum Historica. It's a book one. As I told you, there is a chance of asking books, maybe their discoveries or maybe some other kinds of things. Here, book will be there. So they will ask Rarorium Plantarum Historica was written by Clusius. Okay. Then Robert Hooke. He drawn the sporangia of mucar and teleospores of Phragmidium disciflorum and Phragmidium mucronatum, and he was the first person who observed under microscope of these two pathogens. And he wrote a book called Microscopy in 1665, and he is the first person who invented double lens microscope. I repeat once again, they are not asking microscope. Double lens microscope was invented by. So double lens means having two lenses. Very simple, right? So double lens microscope or compound microscope was first invented by Robert Hooke. It is a very important one. Who invented the double lens microscope, or else who invented the cells? The term cells also introduced by him only. And he wrote a book called Microscopy in 1664. They will also ask like mucar and teleospores of microscopic images of mucar and teleospores was first observed on a microscope by whom? So such kind of things also we do expect, right? Next one is Carolus van Linnaeus. Is a Sweden person. Generally, he's very known, well known for binomial nomenclature, right? He called this ordering of fungi as a scandal of art, and he published a System of Nature in 1735. And he's also produced one more book called Species Plantarum. It's a very important. He's well known for it, right? And he's also considered as father of taxonomy because this is the one who actually started this taxonomy, or the first person who started the classification of fungi or taxonomy. The world's first taxonomist is Carolus van Linnaeus only, and he wrote two books, System of Nature and Species Plantarum, wherein Species Plantarum introduced he thought about around 4,500 to 5,000 kinds of plants. System of Nature also he talked about various kinds of plants and how classification actually works and various kinds of things he have done. So that's why he's considered as father of taxonomy, and he is very well known for binomial nomenclature. And his students are C.H. Persoon and E.M. Fries. I will tell you who these two persons are, but anyway, just remember C.H. Persoon and E.M. Fries are the students of Carolus van Linnaeus. Then next one is P.M. Michelli. He is considered as the father and the founder of mycology. and he wrote a book called Nova Plantarum Genera in 1729. It's a very very important book, and he proved that this is caused by spores, and he described over 900 species of fungi. The three questions we do expect is, who is the father of mycology, who wrote Nova Plantarum Genera. This was a repeatedly asked question in the previous years, if you take a look into. So Nova Plantarum Genera was written by whom, at what year he wrote Nova Plantarum Genera, who proved that this is actually caused by spores. So these 3 points are very very important. Is P.A. Michelli, generally Italian scientist. So one of my professor used to say, the place where they where they belonging to is also very very important. So I suggest you to also remember all those things anyway, but these points are most important things. Next one, C.H. Person. Just now we discussed about C.H. Person, right? He's a student of Carolus van Linnaeus, right? So he's considered as the father of systemic mycology. So classification of fungi through systematic classification of this fungi was started by this person. That's why it's considered as a father of systemic mycology. He's also considered as a father of modern taxonomy of mushrooms, because he started taxonomy giving the classification of mushroom. That's why it's called as a modern taxonomy of mushroom. So his arrangements of mainly based on the spore color, are based on the arrangement of spore color, and he wrote many things like articles of fungi, introduced classification system 1794. The most important thing what he have done is he wrote a book called Synopsis Methodica Fungorum. It is a very 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 important one. Synopsis Methodica Fungorum was written by C.H. Parsoon, 1801. Okay, this is the chief starting point for the naming of fungi. Very very important one. The next one is Charles Darwin, around 1958 and 59. He worked one of the famous theory, that is the origin of species, right? The next one is E.M. Fries. We also discussed that E.M. Fries is also a student of Carolus van Linnaeus, right? So he's considered as a Linnaeus of mycology, which means he's almost greater to Linnaeus after Linnaeus, right? So that's why it's considered as a Linnaeus of mycology, and also considered as father of systemic mycology. This question was asked, like, who is considered as a Linnaeus of mycology, or who is considered as the father of systemic mycology? So before C.H. Person actually started this systemic classification, and later this E.M. Fries had done really significant contributions to this classification. That's why it's considered as a Linnaeus of mycology, and also considered as father of systemic mycology. He also wrote a book called Systema Mycologicum in 1821. This is also a very important book for starting point for the nomenclature of fungi, and he especially dominated Basidiomycota by his approach, that is called Frisian approach. So the question is, who is considered as the Linnaeus of mycology, father of systemic mycology, who wrote Systema Mycologicum, E.M. Fries. So that is a very important thing we must consider. The next one is Matthew Dutillett. He is considered as a great grandfather of phytopathology, or great grandfather of plant pathology. So he worked on generally contagious diseases like burnt wheat. At the time, he told that this burnt disease is contagious, and also recommended some kind of salt solution for the management. But he is generally considered as great grandfather of phytopathology. In 1840s, Tulsen brothers, L.R. Tulsen and one more Tulsen, both are brothers. Actually, they are considered as a reconstructor of mycology, because so many discoveries that they have done, they have all put together in an order, and make the mycology into an exact order. That's why they considered as a reconstructor. They have actually reconstructed all the scattered things together. That's why they considered as a reconstructor of mycology, and they are the one who recognized the 5 spore stages of Artosius rust in 1854. And they wrote a book called, not a 5 spore stages, like couple of spore stages, because not all 5 spore stages was discovered by them, because the 5th stage, that is the zero stage was discovered in 1927 by Craig, right? So this normally they have recognized some almost not 5, that was I can say for sure. And they wrote a book called Selecta Fungorum Carpologica. This is also a very important one, and they wrote in between 1861 to 1865. And J.G. Kuhn, he is from Germany. He published the first textbook on plant pathology, that is the diseases of cultivated crops and their causes and their control, in 1858. This is a very 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 important question. It's been many years that I am reading this question, like who wrote the first textbook in plant pathology, that is the diseases of cultivated crops and their causes and their control, in 1858. That is J.G. Kuhn. And who they will ask, like they gave the book, and they may ask like who wrote this, such kind of of things will be there, right? So it is a very, very important one. You remember the book name, The Diseases of Cultivated Crops and Their Causes and Their Control in 1858. If you want, you can take a notes, as I told you in the previous preparation video. Just take your own notes, that will be very easy for you. And considered as a microscopic manager, generally considered as a microscopic manager, because he used to work on microscope a lot, so that he was well versed in microscope. That's why he considered as microscopic manager. The couple of book, it is also mentioned that he is considered as father of plant pathology, but we know very well that Anton de Berry we are considering as a father of plant pathology because of its the largest contribution that he have done to the plant pathology, right? The next one is M.J. Berkeley. He's the first person who actually tried to prove that lead blight is caused by some kind of fungi. But anyway, he's the first person who fought with John Lindley to prove this is caused by actually fungi or some kind of things, right? Okay, and he's the first person who, I mean, he's the one who coined the term mycology. We know mycology. What is mycology? Study of fungi. Very simple. Systemic mycology, and he have systematized, identified around 6,000 species, and he published two books: Introduction to Cryptogamic Botany 1857 and Outlines of British Fungology in 1860. He is one of the very important person when it comes to British mycology. So he began the publications on vegetable pathology in 1854, and he is the one who gave Idium tuckeri to the grapes powdery mildew. Now we know it is caused by Uncellina necator or Erisife necator. At the time he gave this is caused by Idium tuckeri, and exams in exam we can expect questions like this. Who is considered as, I mean, who coined the term mycology, or who gave grapes powdery mildews caused by Idium tuckeri? Such kind of things we do expect, right? Now coming to the modern era, Henry Chanton de Berry 1831-1888. I would say 1831 January 26, 1888 January 19, I think. Anyway, he's a German surgeon. He's originally a doctor, but interest was in botany, so slowly shifted to botanist and later become the greatest mycologist. Right? He's largely considered as a father of plant pathology, father of modern plant pathology, and father and founder of modern experimental plant pathology. And he wrote books like a Comparative Morphology and Biology of the Fungi, Mycetosia, and Bacteria. And I also gave lectures on bacteria 1885, on mildew and fermentation 1872, Comparative Anatomy and Vegetable Organs of Phanerogamic Fams. And the most famous book is Morphology and Physiology of Fungi, Mycetosia, Bacteria, that was written in 1866, but English translation happened in 1887. That was actually translated by E.F. Garcia and revised by Isaac Bayflor. Anton de Berry introduced terms like he's the one who introduced terms like Hastoria, otherwise called as symbiosis, teletospores, mycetosia, sex hormones, iron anthradiol, autoceous and heteroceous rust, and chlamydospore. And his students like M.S. Voronin, who discovered the life cycle of clubroot of cabbage, and Oscar Brefield, pure culture technique, A. Milladet, PMA Milladet, Bodo Mitcher, H.M. Ward, tropical plant pathology, W.G. Farlow, Fisher, and all are later become the greatest contributors. That was all are trained by Anton de Berry only. Anton de Berry, well known for the development or discovery of the life cycle of Phytophthora infest that is caused by lead blight of potato Irish famine, and he demonstrated the heterogeneous nature of stem rust in 1865, and is also the first to work on physiological plant pathology, work on botrytis cinerea, and he also the one who recognized the role of toxins in direct penetration by tissue disintegration, and he also sclerotia, the tissue disintegration into sclerotia in 1886, and he is the one who named this Phytophthora infestans. Phytophthora means plant destroyer. Because of his greatest contributions, he became renowned as the father of plant pathology. He is Heinrich Anton de Berry from Germany. The next one is M.S. Voronin. Just now I told you Voronin is a student of Anton de Berry, and he discovered the clubroot of cabbage life cycle that is caused by Plasmodium brassicae. See, you just consider an exam point of view. Okay, who discovered the life cycle of clubroot of cabbage? Voronin. What is the life? Clubroot of cabbage is caused by Plasmodium brassicae, and also the one who founded the life cycle of potato wort, which is caused by Synghytrum endobacterium in 1878, and also discovered the Voronin boys. He is Voronin. This is a clubroot of cabbage symptoms. H.M. Ward is also a student of Anton de Berry. He is considered the father of tropical plant pathology. Just now I told you. Emphasis on role of environmental epidemiological factor on coffee rust that was given by this H.M. Ward. Actually, I will tell you the small history. At the time when coffee rust happens in Sri Lanka 1870, British wrote a letter to Anton de Berry to help us what exactly going on. So Anton de Beer at the time, he was very busy with other activities, so that he sent a letter back to British government that he is sending one of his students, that is H.M. Ward. H.M. Ward went to Sri Lanka and identified what exactly the reason behind it. He come to a conclusion that the main reason is epidemiological factors, first one. The second one is uh, continuous cultivation of, I would say, monocropping. Very simple. I will finish it with monocropping. So, and later he identified the life cycle and all the things that was given by him. And we, remember 2 points: father of tropical plant pathology and the life cycle of coffee rust was given discovered by H.M. Ward. Okay. And then Millard, just now I told you, is also a student of Anton de Barry, and he discovered Bodo mixture for the control of grapes downy mildew. It is a very, very important and repeatedly asked question. Who discovered Bodo mixture? PMA Millard. Bodo mixture is recommended for the management of which disease? The grapes downy mildew. Bodo mixture is originally developed for which disease? Grapes downy mildew. So it is also called as serendipity discovery, accidental discovery. Okay. The composition of Bodo mixture is generally, the composition is 1:1:100, but it is not the exact composition that Millard give. But if you see in a overall way, this actually matches the composition, that is 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, 100 liters of water. As I told you, the original formula was given by 5 lb of copper sulfate, 5 lb of lime, and 50 gallons of water. When you calculate in that way, also it comes close to 1:100 only. So he named this borax. It is actually the university name, and the area where he discovered is also the borax name. That's why to honor it, he named it as Bodo mixture. Okay. Next one is Erickson. He described the phenomenon of physiological specialization of wheat in wheat stemras. So, like who discovered or who identified or described the phenomenon of physiological specialization? That way also we can see. Erickson, 1894. Okay. Next one is P.A. Secado. He's also very important one, or systematis. He's a professor at Pada University, and he's well known for spore grouping of Ascomycota, that is sac fungi and Tetromycota, dustbin fungi and fungi imperfecti. And his famous book was Silloge Fungorum or Sillum Fungorum, wrote in between 1882 to 1831. Around 26 volumes will be there. 25 volumes, but originally 26 volumes. And the full name of it was Silloge Fungorum, or Silloge Fungorum Omnium Huscum Cognitorium. And he also wrote other books like Fungi Italic, that is published between 1877 to 1886, and appeared in around 38 volumes. The most important thing is Silloge Fungorum was written by F.A. Secado. Okay. J.F. Daster is actually Indian scientist. We might have discussed. We will also discuss in Indian related scientists. Okay. Anyway, let me tell you, he's the first president of Indian Pathological Society. Actually, he's supposed to be this. He's actually the second president. First president is S.R. Bose. Okay. He's the second president of Indian Pathological Society. He's a well-known, renowned for the establishment of Phytophthora genera in India in castor seedling blade, that is called Phytophthora parasitica. And it's not first president, it's second president. Okay, in 1940, around 48. And is also the one who first reported the first viral disease of India, that is sugarcane mosaic virus. That is also first reported by him only. And the questions repeatedly asked question is, who established the Phytophthora genera in India? J.F. Daster. Seedling blade of castor was actually associated with which of the following scientists? J.F. Daster. Actually, seedling blade of castor was first discovered by J.F. Daster only. Next one is E.J. Butler. He is generally considered as father of plant pathology in India, and is also the first empirical mycologist in India. Because of its largest contribution that they have done to the plant pathology in India, he is largely considered as father of modern plant pathology in India, and also the mycology in India. He is the first director of Empirical Bureau of Mycology in England. He wrote a book called Fungi and Disease in Plants. In fact, he wrote many books, but this book is very famous, Fungi and Disease in Plants, 1980. And he also wrote monograph on Pitheaceous and allied fungi, the fungi in India, along with the Guy, Richard and Bisbee, 1931, Plant Pathology with S.G. Jones. And he is the one who erected the genus called Allomyces. So the genus Allomyces was created by E.J. Butler, who is considered a father of plant pathology. E.J. Butler. First Empirical Bureau of Mycology was first director. Who is the first director? Who is the first empirical mycologist in India? Such kind of questions we would expect. He's also discovered the first plant bacterial disease in India, bangle blight of potato. And I was a little bit doubted about, because I have gone through various kinds of resources, and it mentioned that the first plant bacterial disease was reported by Capel 1892, something like that is mentioned in various areas, but many of the things it is mentioning that it was first plant bacterial disease, that is bangle blight of potato, was discovered by E.J. Butler. Anyway, majority of the sources are saying first plant bacterial disease was reported by E.J. Butler only. Go with this, and these are all the couple of questions we would expect from Butler. Then we see Stockman. The term hypersensitive was coined by E.C. Stockman only, and later he also demonstrated a physiological stem rust in 1917 during his PhD time, and also studied the air dispersal of rust fungus in 1920. The most important thing is hypersensitivity was first coined by E.C. Stockman 1915. JH Craig, as I told you, the fifth zero stage, I would say. Actually, the zero stage that actually connects the other stages, that was first discovered by JH Craig in 1927. We can also call it as zero stage or pycnial stage or spermatial stage or sexual stage of wheat stem rust fungi. And it's also discovered heterothallism of Puccinia graminis to reveal this function of sexual organ. And this is about JH Craig. It's a very important person related to wheat stem rust. Alexander Fleming, he isolated the first antibiotic in the world, that is penicillin, isolated from a fungus called Penicillium notatum. Now this Penicillium notatum is called as Penicillium chrysogenum. So for his discovery, he shared Nobel Prize with Ernest B. Chain, Harvard Walter Florey in 1945. So anyway, I'm telling you, just there are find of facts. There are a lot of things to look into. They will ask questions like who identified the first. I'm going a little bit faster. Hope you guys understand because we have a lot of time to to read all those things. So we cannot waste the time here itself. So that's why I'm just telling the facts and all those things. Anyway, the first antibiotic was isolated as penicillin, isolated from Penicillium notatum, isolated by Fleming. And there are also questions like he shared a Nobel Prize with whom? B. Chain and Albert, Walter Flor and Ernest B. Chain and Howard Walter Flor in 1945. But is actually discovered in 1928 and published the paper in 1929 around August time. So this is about, in such a way the questions will come. I'm just telling the kind of important facts and all those things. So hope you guys understand. The next one is J.C. Walker. He identified catechol and protocachic acid are the resistant factors in onion against onion smudge disease. Generally, there is a phenomenon called phenolic theory, which the theory says that red onions are highly resistant to onion smudge disease. They will ask, red onions are resistant to which disease? Onion smudge. Onion smudge is caused by Collotrichum circinans. What are the compounds that is present in the red onions that become resistant against this onion smudge disease? Catechol and protocachic acid. In such way they will ask. Okay, what is phenolic theory means? In red onions, they are highly resistant to onion smudge disease because they contains two phenolic compounds called catechol and protocachic acid. These two acids are absent in white onions. That's why white onions are highly susceptible to onion smudge disease. That is the only thing that was given by J.C. Walker. And he also wrote one monograph, I think, on tomato, some kind of experimental on tomato, something like that. I don't know exactly, probably. And he's also developing resistant varieties related to onion, cabbage, beans, potatoes, beetroot, and cucumbers. Muller and Borger, they were the first who coined the term phytoalexins, and the first phytoalexin is also coined by the first phytoalexin is a pisatin. That was also given by this Muller and Borger. That is isolated from pea plant. Okay. Next one is Paul Niergaard is generally considered as father of seed pathology. They will ask who is father of seed pathology. Who coined the term seed pathology? And Paul Niergaard, along with one more scientist, Mary Nobel, they coined the term or introduced the term seed pathology. He also wrote a book called Seed Pathology. It's how coincide it is, right? So he's the father of seed pathology. He's the one who coined the term seed pathology, and he wrote two books on seed pathology, volume 1 and volume 2, that was published in 1977. Year is also very, very important. And he's the first chairman of ISTA, International Seed Testing Association, between 56 to 74. H.H. Floor is a very important person. I will say a very simple thing like a gene for gene hypothesis was given by him while working with the linseed rust, which is caused by Melamsora lini, and he is the one who proposes the term A virulence gene also. The gene for gene hypothesis simply says for every resistant gene in the host, there must be a corresponding and A virulence gene in the pathogen. That is called gene for gene hypothesis. In exam, what kind of questions you would expect is gene for gene hypothesis was given by H.H. Floor in the year 1946 while working with witch rust, linseed rust, or we can also call it flaxseed rust. Linseed rust or flaxseed rust is caused by Melamsora lini. Who coined the term A virulence gene? H.H. Floor only. These are all the various kinds of questions. I'm just telling the model. Okay. Next one is Ponticorvo and Roper. They are well known for parasexuality. Their mechanism called parasexuality. Generally, majority of the fungi reproduce baby sexual or asexual, but couple of fungi like Dendromycota group of fungi they reproduce by means of another mechanism called parasexuality, which means plasmogamy, karyogamy, meiosis will not occur at a regular stage or a specific point of manner. They will occur like whenever like not in a regular manner. So that is called parasexuality. Was first discovered by Ponticorvo and Roper. Here is also very important. 1952. They discovered in a fungi called Aspergillus nodulans, now called Vermicella nodulans also. J. Vanderplank. Regarding this person, we will discuss in epidemiology. Anyway, I will tell you, he is generally considered the father of plant disease epidemiology. He gave the concept of vertical and horizontal resistance, and he is the one who introduced the term vertifoliar resistance to describe the horizontal resistance. And he published many books, and the very famous book is Plant Disease Epidemics and Control, very important one, 1963. And other books include Disease Resistance in Plants 68, Principles of Plant Infection 75, Genetics and Molecular Basis of Plant Pathogen 78, Host Pathogen Interaction in Plant Disease 1982. Norman E. Borlaug, we know this person as the father of Green Revolution, and he got Nobel Prize in 1970, Nobel Peace Prize, and he developed semi-dwarf fire-resistant wheat varieties. He did his, he was well versed in genetics, agronomy, plant pathology, and all the kind of things. And just remember one thing: the father of Green Revolution. Just to honor him, I have placed this, but is not that much related to plant pathology. I would say he's much more geneticist than plant pathologist. Anyway, he have worked on both aspects, that is secondary. Next one is M.J. Thirumulachar. He's well-known. Generally, he's also considered the father of Indian antibiotics. Indian antibiotics father. Okay, he's well-known for the development of antifungal antibiotics called Ariofungin and other antibiotics like hyamycin, dermostatin, MYC4, tetraenion. All the antibiotics was developed by him only. He wrote a book called Antibiotics and Plant Disease Control. And he also wrote monographs like Ishnoagenils in India, Irionels of the world, Cercospora, Physoderma, and Cephalosporium. These are all the various kinds of things that he have done. Jacob Cotton, he is considered as father of soil solarization. He developed a technique called soil solarization by using a thin layer of polythene sheet. We can heat the soil so that the soil-borne pathogens can be dead. It is only recommended for soil-borne pathogens, and the suggested size of polythene bag is 100 microns. Dickman and Collectcode in 1989, they successfully transformed a wound infection fungi that is actually coding a gene called cutinase for cutinase for the cutin degrading of cutin, and also demonstrated the function of enzyme cutinase that is actually responsible for the direct heat or direct penetration. And this cutinase form Fusarium solaniforma species PC that cloned and actually the first sequenced. Next one is classic Ruskin's matrix on royals. They demonstrated the salicylic acid that is systemic acquired resistance. And De Broglie and Chen, they also transformed a chitinase. They, Digman and collected cutinases. Here Broglie and Chen, they transformed chitinases enzyme that exhibit actually resistance, increase the disease resistant to tobacco. Okay, and they identified this sylvine synthesis, the enzyme which is actually synthesizes the phytoloxin, and it exhibits the enhanced disease resistance in tobacco plants. DeWitt, he's the first person, it's a very important one, cloned or isolated the first fungal avirulent gene, that is Cladosporium fulvum. Briggs and Walton cloned the first resistant gene HM1. I repeat, first fungal avirulent gene is Cladosporium fulvum, first resistant gene is HM1. And Dean et al., he's the man who actually first completely genome sequenced the plant pathogenic fungi, that is Magnoporthe as a rice blast. So rice blast was sequenced by R18. What is the first plant pathogenic fungi to be completely genome sequenced? Magnoporthe graciae. So such kind of things we would expect. Now we will see the historical development of plant pathology in India. The first one is JF Daster. We have already discussed about JF Daster. So without wasting any further time, we will skip the scientists that we have already discussed, and we will discuss about the new one. T.S. Sadasivan, he is generally considered as father of hypomycetes, and he developed the concept of vivotoxin. So the concept of vivotoxin was developed by T.S. Sadasivan, And actually, the term vivotoxin was given by Diamond and Wagner, but the concept was given by the C.S. Sadasivam only. And he is the first person who demonstrated this fusaric acid, that metabolite that is produced by the fusarium species, especially in wilting of cotton, which is caused by fusarium oxysporum, pharma species vas infectum. And is also the founder of School of Physiological Plant Pathology at Madras, and his contributions on taxonomy of hypomycetes, and that's why he is considered as father of hypomycetes, right? K.C. Mehta, he is considered father of Indian rust, and he discovered the life cycle of wheat stem rust in India. And is also reported that couple of things it is mentioned that it is reported the alternate host Barbary, but it is saying that K.C. Mehta not reported this Barbary as alternate host, and he has actually doubted on it. But now I believe that maybe there is a chances couple of articles is mentioned like that, couple of articles it is mentioned in a reverse way. Anyway, we must sort it out what exactly the truth is. Anyway, the life cycle was wheat stem rust in India was first discovered by this K.C. Mehta and published two monograph further studies on the cereal rust in India. The part 1 at 1914, part 2 at 1952, and he established three laboratories in Agra, Almore and Shimla. The B.B. Munkur is a very important person when it comes to the Indian plant pathology, and he started the IPS, Indian Pathological Society, and he issued the first journal, that is Indian Pathology, in 1948. And also wrote a book called Fungi and Plant Disease in 1948, not 49, it's 48, okay. And Fungi and Diseases in Plant was written by E.J. Butter, but Fungi and Plant Disease was written by B.B. Munkur. Nine, he reported the rice cera disease. We used to study. This is one of the standard, like hallmark question, right? So rice cera disease is caused by zinc deficiency. It was first reported by Nine. He authored a book called Fungicides and Plant Disease Control, Nine and P. Anthopleal, and he also worked on the PGNP. D.D. Sinningham, he proposed a new genus called mycologia, generally the plant parasitic algae that he placed under this, and he reported a new algal disease that is called red rust of tea, which is caused by Cephalourus virescens. There are three important diseases that we used to study that is caused by algae: red rust of tea, red rust of mango, and red rust of guava. All these three are known to be caused by Cephalourus virescens. In 1899, he identified this causal organism that causes are responsible for this red rust of tea, that is Cephalourus virescens in Assam. K.C. Mehta, I think we might have discussed earlier about this K.C. Mehta. Anyway, he is generally considered father of Indian rust, and he studied about epidemiology of wheat, cereal rust, and all those things. And the most important thing is he showed that Barbary is an alternate host for wheat stem rust. He is the first person who showed that Barbary act as an alternate host, and he wrote one monograph, further studies on cereal rust in India, part 1 and part 2. Part 1 is probably 1940 or maybe 48, or second part is around 1952. And next one is S.R. Bose. He is the pioneer who actually works on bracket fungi, polyporales, and he is very famous for. He is also known for his. He is the first president of Indian Pathological Society. Second president is JF Dash, because he is the first president. And along with the B.B. Munkur, he collaborated with the B.B. Munkur for the establishment of Indian Pathological Society. Next one is the R.S. Singh. He is generally considered as a lighthouse of Indian plant pathology. His written books are, he wrote many books, and those books are the widely, read books like plant diseases. Generally, we consider as Bible of Indian plant pathology. Principles of plant diseases, disease of vegetable crops, disease of fruit crops, plant disease management, a handbook of economic nematology. These are all the different kinds of works that was done by R.S. Singh. Now we will see about bacteriology. Generally, the term bacteria was coined by Ehrenberg, but in 1676, we used to study something like Anton van Leeuwenhoek was the first person who identified or who discovered the bacteria. Of course, he is the one who discovered the bacteria, but the name was given by Ehrenberg in 1838. So starts with Francesco Redi. He is generally considered as the father and founder of experimental biology, and also considered father of modern parasitology. He is the first person who disproved this spontaneous generation theory. At the time, people used to believe that spontaneous generation theory because organisms are arised from an inanimate organism, something such kind of belief was there in that system. But he is the first one who disproved this spontaneous generation theory, and by conducting the experiments on the generation of insect and published in 1668, and he told that these insects are actually came from eggs or flies, not from the inanimate agents and all those things that was told. So this is the first proof of disproving the spontaneous generation theory. Next one is Anton van Leeuwenhoek in 1675 and 1676. He is the first person who developed a microscope, is also considered as the father of plant bacteriology, and he developed hundreds of microscopes, and along with the most important thing is the magnifications. He obtained the magnifications up to 50 to 300, and he published his first publications on bacteria in 1683. And he also discovered spermatozoa in red blood cells, and these are all in exams of how we can kind of questions we would expect is like who is father of bacteriology, and who developed magnification lenses for microscope and all those things. That was in previous we might have discussed about Robert Hooke. Double lens microscope was invented by Robert Hooke, right? The next one is Louis Pasteur, very famous man. He is generally considered as the father of modern bacteriology and the founder of modern microbiology, and the famous theory that was given by his germ theory of disease, which completely demolished the spontaneous generation theory. He is the first person who experimentally proved with 100% proof that spontaneous generation theory was not right, and initiated the germ theory of diseases. And he developed principles for vaccination, microbial fermentation, and pasteurization. We know very well this is very well known for his pasteurization techniques, and also created the vaccine for rabies and anthrax. And pasteurization, generally what is that? Generally heat killing of microorganisms. For wine and beer it requires 57 degrees Celsius. For milk it is 62.8 degrees Celsius. Very very important one. What is the pasteurization temperature? 62.8. Generally we general pasteurization temperature, and it also goes high and goes down along with the depends on the beverage we are using. Next one is Robert Koch. He is also a very famous man. He is considered as a father of modern bacteriological techniques and microbiological techniques, and he is also considered as father of modern founder of modern bacteriology. And he proposed Koch postulates and pure culture techniques. And this Koch postulates is also known as pathogenicity rules, which are very important to prove the pathogenicity of any organism, mostly fungi, bacteria, but not applicable for virus and virides, but mostly fungi and bacteria. E.F. Smith, he gave the first proof that bacteria could cause plant disease. Before that, when actually the first proof was given by not E.F. Smith, the first proof was given by T.J. Burrill in 1878. He discovered a first plant bacterial disease called fire blight of apple, which is caused by Erwinia amylovora. So that is the first proof, and later he also proved that bacteria can cause plant diseases. And he worked on bacterial blight of cucurbits in 1996 and crown gall. Those couple of works he have done. So that's why he's largely considered father of phytobacteriology. And his books includes bacteria in relation to plant disease in 1905, 1911, and 1943 volumes are published. And introduction to plant bacterial disease in plants is in 1920. So actually, among the four Koch postulates, there are four Koch postulates are there, right? So first three Koch postulates was given by Robert Koch, and the fourth Koch postulate was given by E.F. Smith. And in 1877, Nageli actually suggested the pleomorphic bacteria, and pleomorphism in bacteria just changing their shapes, right? And S.D. Dillett is generally a retired surgeon who coined the term microbe. T.J. Burrill, as I told you, founder of phytobacteriology, because he identified the first plant bacterial disease to cause plant diseases. That is a fire blight of apple, which is caused by Erwinia amylovora. So the previous name of Erwinia amylovora is Micrococcus amylovora. Later name changed to Erwinia amylovora. Christian Gram, he developed gram staining technique to differentiate gram positive and gram negative bacteria. Twart and Herrelli, who discovered bacteriophages. So the bacteriophages were individually discovered by Twart and Herrelli, but the term bacteriophage was given by Herrelli. In 1923, Bergey's Manual for Determinative Bacteriology, which was started by David H. Bergey, which is a non-profitable organization. So this Bergey's Manual mainly deals with the classification and all the stuff, and new kinds of updates, classification, everything will be mentioned in this Bergey's Manual of Determinative Bacteriology. In 1925, Johnson and Mervyn actually the first demonstrated the role of toxins, that is especially wildfire toxin in tobacco. Wildfire toxin, that is wildfire of tobacco, is caused by Pseudomonas syringae, Pathovar tabaci. In exams, they will ask who first demonstrated or what is the first toxin to be identified, or some kind of things that we can expect, right? In 1925, Kuhn and Cotilla that isolated the bacteriophages of Bacillus keratovirus. Selman Waxman is largely considered as the father of soil microbiology and also considered as father of antibiotics. Father of Indian antibiotics is M.J. Tirumalajar, but generally father of antibiotics means Selman Waxman. He discovered one of the most phenomenal discovery, that is streptomycin. It is an antibiotic which is isolated from a bacteria called Streptomyces griseus in 1943. For that he got Nobel Prize in Physiology and Medicine in 1952. Lederberg and Tatum. So they discovered a bacterial phenomenon called conjugation. It is a kind of sexual recombination that was discovered in Escherichia coli, and he also coined the term plasmid. And for that they got Nobel Prize in 1958. Clement, so recognized this hypersensitive response in bacteria. So the hypersensitive response in bacteria was first identified by Clement. That hypersensitive response means programmed cell death. Imagine this is a leaf. So the adjacent cells of the leaf will die to prevent the further spread of the infection. That is called uh, programmed cell death or hypersensitive reaction. And the next one is Chilton. He introduced DNA plasmids of Agrobacterium tumefaciens into the cells of higher plants. Next one is Taskovic's first A-virulent gene. He included the first A-virulent gene from Pseudomonas syringae, pathovar glycinia. So the first bacterial A-virulent gene was cloned by Taskovic's in 1984. And Linden Green, Peat, and Ponopolus, they discovered the hypersensitive response in the HRP genes, that is the Pseudomonas syringae, pathovar fasciola. And Kereni and Staskovic's A-virulent gene of Xanthomonas campestris, pathovar campestris, AVRBS2, act as an effector for the pathogen A-virulence. And Pawlowski, he was the first person who identified this Xanthomonas can encode gene clusters that is actually associated with Xanthomonas campestris. And in 1997, actually determined there is a one gene called PIGB. So he identified the gene that is actually non-pigmented gene which gains partial pigmentation, the strains of Xanthomonas campestris, Pathovar campestris. And Mills, he developed the PCR ELISA, especially for the detection of Ringrata potata, which is caused by Clavibacter michiganensis subspecies Epidonicus. Klepper is generally considered as father of PGPR, plant growth promoting rhizobacteria. Van Loon, he's a very known, well-known, famous for his PR proteins, pathogens related protein. In one of the paper that was published in 2006, he have mentioned around 17 groups of PR proteins, right? Like PR1, PR2, PR3. There are various other kinds of PR proteins, right? Like that. In 2008, Baker, he wrote a book called New Perspectives and Approaches on Plant Growth Promoting Rhizobacteria. And Andre Munzo, the screened the cry, weep, sipped genes in Bacillus thuringiensis. These are different kinds of genes that is responsible for various characters. Now we'll see virology. Virology will start with the 16th century, tulipomania, tulip color breaking. At the time, tulips got very good color variations or some kind of petal variations. At the time, people used to believe that it might be some other reason, but they got very good price just because of this petal variations. In later stages, it was identified as a potivirus and known to be transmitted by aphids. Adolf Meyer, the virology was actually started with this persons like Adolf Meyer, first one who actually coined the term mosaic. So they were he's the first person who started working on mosaic disease of tobacco, and he coined the term mosaic and also proved that transmissible nature of tobacco mosaic virus, and he called that sap transmission of disease or that sap transmission is known as mosaic concrete, which is a German language. Okay, and they later performed various kinds of experiment and identified that filterland paper and even through the virus particles retains infectivity, because at the time he don't know whether it is a virus or something else. So after performing the Chamberlain filter experiment, Chamberlain filter experiment is what? So just filters the bacteria, because he thought that it may be caused by bacteria. So that's why he performed Chamberlain filter paper. Still after Chamberlain filter paper, still they got the infection. So in exams we can expect like who thought that tobacco mosaic disease was caused by bacteria? Adolf Meyer. Who coined the term mosaic? So such kind of diseases. Hope you guys, I'm just giving the hints. Next one is Ivanovsky, is from Russia, the first person who actually discovered the inclusion bodies, and the most important point is concluded that after performing the Chamberlain filter experiment, he thought that bacteria actually, if it is after performing the Chamberlain filter experiment, even after performing this, the disease still occurs, which means the causal organism is smaller than that of bacteria, but it is not caused by bacteria. Such kind of things he have told. So and also inclusion bodies he have discovered. Next one is Bijgernik, very important person when it comes to virology. He is a general Dutch microbiologist and botanist. He is largely considered as father of plant virology and also considered as father of environmental ecology. So after performing various kinds of experiment, especially agar diffusion experiment, and he coined the term virus. The term virus is a Latin word which means venom or poison, and he called that a liquid material because, as I told you, it is transmitted by sap transmission. Who told it is sap transmission? That Adolf Mayer 1886. So that sap material is, he called it as contagium vivum fluidum after performing various kinds of experiment and repeated, and he come to a conclusion that tobacco mosaic disease was caused by virus. That is the infectious liquid material is contagium vivum fluidum. And he's also discovered various other kinds of things like spirillum destructans are now called as desulfobacterium desulfuricans. Clostridium bigernicki is a gram-negative bacteria which was named after this bigernic. Twart and Harely, we might have discussed the first person who discovered bacteriophages individually, but the term bacteriophage was coined by Harely only. Holmes, they performed local lesion assay. So the local lesion assay was performed by Holmes. Very simple local lesion assay. Generally, the amount of virus present on the plant sap is directly proportional to the amount what you are rubbing and what other plant is getting. That is actually local lesion assay. And they performed in two hosts are called Nicotiana glutinosa and Nicotiana tabacinem. He actually performed this experiment. The most important thing is local lesion assay. They will ask who performed a local lesion assay. Holmes. Next one is McKinney. In 1929, there is another phenomenon was discovered by McKinney, that is a cross protection. We can also call it as a pre-immunization. They actually developed originally, the cross protection technique is originally developed against tobacco mosaic virus, but later identified this cross protection is very effective against the citrus trichisa virus, right? So what is cross protection? Just pre-immunizing the plants to protect from the severe strains of the plants. So pre-immunizing can be done with mild strains of plant viruses to protect the plant from severe strains of plant viruses. So this is a mechanism. Nolan Ruska, they were the first person who invented this electron microscope. So electron microscope was invented by Noll and Ruska. So the Ernest Ruska and Noll in 1931, they developed a microscope which is having a very powerful, which can capable of magnifying 400 times. And later Ruska alone started working on it, and the magnification was gone to 1 lakh, and later we know the history, like lots of microscope we do have that can even now we can magnify up to 10 crores, right? So then Ruska built a first electron microscope, though they will work on a principle called high resolution and optical microscope, and for that Ruska got Nobel Prize in 1986. K.M. Smith, he was the first person who thought that use of indicator plant in viral diseases, and he is also the first person that separated potato virus and named as X and Y, and he wrote a book called Plant Viral Disease in 1937. And Martin Schlesinger, he is an Hungarian chemist, he is also a very important person when it comes to virologist. Actually, he separated this and purified the bacteriophages by using a technique called ultracentrifugation, and the later he identified this bacteriophages contains equal amount of proteins and DNA that was given by Martin Schlesinger. W.M. Stanley is also a very important virologist, and he done the purification and crystallization of plant viruses, especially the TMV. Questions will be like, who purified or who crystallized the tobacco mosaic virus? W.M. Stanley. What is the chemical used for the crystallization? Ammonium sulfate. This is a very important, ammonium sulfate. And he received Nobel Prize in 1946 for the crystallization of tobacco mosaic virus. And molecular plant pathology work is also initiated by this W.M. Stanley. But he thought that this nucleic acid is virus is a nucleoprotein, but he thought that protein coat is the infectious material, but later Geier and Scram identified protein coat only protect the nucleic acid, and nucleic acid is the infectious material. If you see the virus, this is the virus, inside nucleic acid will be there. What W.M. Stanley thought, this protein coat is the infectious material, but later Geier and Scram identified this nucleic acid which is present inside is the infectious material. FC Bowden and Pyrie. In fact, the Nobel Prize is supposed to goes to this Bowden and Pyrie. They have done lots of significant contribution. These are the first person who told that virus is a nucleoprotein. The term first was given by them only. So virus contains both nucleic acid and protein. That's why named as nucleoprotein, the combination of nucleic acid and protein. And they also tried purification of TMV in 1936, but somehow they got a little bit lagged behind this W.M. Stanley. Later W.M. Stanley purified, and after that they also purified, and they come to a conclusion that virus can be purified, and so many things are done. And they used they used phosphate to detect the inheritance of RNA molecule and other kinds of things will be there. But in exam we do expect like who called virus is a nucleoprotein, Bowden and Pyrie. And RDNA, they were the first to build a scanning electron microscope. The next one is Cauchy. Cauchy is the first person who saw the virus in electron microscope. The electron microscope, for the first person who saw the virus in electron microscope is Cauchy. And next one is Delbert and Ellis. They worked on the replication cycle of bacteriophages. Luria and Delbruck, they discovered the bacterial mutations. In bacterial mutations was discovered by Luria and Delbruck. Then you can ask me why these people are there in the virology. Of course, Delbruck alone discovered the mutations in virus. So bacterial mutations was discovered by both, but the Delbruck alone discovered the mutations in virus, and they considered as the founding fathers of phage genetics, and they received Nobel Prize in 1969. And Watson and Crick, we know very well, they discovered the double helical structure of DNA, deoxyribonucleic acid. Actually, this, of course, this credit also goes to someone else. One more person, she got, she almost discovered the DNA, but she couldn't able to identify what exactly it is. But I forgot her anyway, actually I forgot her name, but she is the original lady behind this double helical DNA identification. But later Watson and Crick got quickly catches their thing, and explained that DNA is a double helical and all those things. They were explained in 1953 in between 1956, and they got Nobel Prize in 1962. Hersey and Chase experiment. So they demonstrated the infectious behavior of bacteriophages and also showed that how the DNA is entering inside the plant cell. This is the bacteriophage, right? So how the DNA is entering, all the mechanism they have shown. This is also one of the very important experiment in plant virology. And Geiger and Scram, we might have discussed these persons, right? So what W.M. Stanley thought, this protein coat is the infectious material, but later identified nucleic acid is the infectious material. The nucleic acid is the infectious material was first given by Geiger and Scram. It's a very, very important question. If you see, take a look at the 2018 and 19 question papers, this question was there. Who proved that nucleic acid is the infectious material? Geiger and Scram, 1956. It is a very, very important one. They also investigated about the double-stranded RNA viruses. Hewitt et al. The first person who reported that grape family virus is known to be transmitted by Ziphinema index, that is a nematode. Tickle first reported the fungal transmission. Schiffer first reported the first first DNA viruses, that is a cauliflower mosaic virus, which consists of double-stranded DNA as a genome. Takabe and Wotsuke, they have described the techniques for the propagation, infection of protoplasts of viruses and all the kinds of things. In 1966, the International Committee on Nomenclature of Viruses, it is a very, very important, ICNV, International Committee on Nomenclature of Viruses. Later, the name is changed or shifted to International Committee on Taxonomy of Viruses in 1973. Kohler and Milstein, they developed a hybridoma technology for the production of monoclonal antibodies. They who developed the production of hybridoma technology for the production of antibodies, such kind of questions you would expect. Kohler and Milstein. Wohler, ELISA, of course, Wohler, Clark and Adams, they all are related to ELISA only, but mostly majority of the books we do study Clark and Adams. The technique developed the ELISA technique and used the ELISA plate for plant virus detection. In the internet, we do get two names like Peter Perelman and Eva Evangel. They were the first two actually worked, but this technique was purely developed by Clark and Adams. Aaron Klug, crystallographic electron microscopy, he was the first one who developed that crystallographic electron microscopy for elucidation of nucleic acid protein complexes and all the things. Paul Abbe and Beachy et al. They initiated molecular plant pathology while working with the tobacco mosaic ware, and they also demonstrated that first transgenic gene that is actually associated or mediated with the coat protein mediated genes, resistance. So this coat protein mediated gene resistance was first developed by Beachy et al. Stanley Prusiner, he discovered prions. Prions are nothing but infectious proteins or malfunctional proteins, I would say. For that, he got Nobel Prize in 1997. Andrew Fire and Craig Mello, this is one of the revolutionary discovery. They discovered RNA interference, RNA silencing. We can also call it as RNA interference or RNA gene silencing in a nematode called Chinorhabdis elegans. We can also call it as post-transcriptional gene silencing as well. For that, they got Nobel Prize in 2006. So in questions, we would expect like this. What is the nematode associated with RNA interference, or otherwise called as post-transcriptional gene silencing, such kind of questions. Chinorhabdis elegans, nematode, RNA interference, also known as post-transcriptional gene silencing. Now we'll see epidemiology. NACOB first devised the a scale to measure the plant disease intensity. Erickson demonstrated the physiological resistance in fungi. H.M. Ward, he is the first person actually started studying diseases towards the epidemiological factor. When he went to Sri Lanka while working with coffee rust, he was the one who started exploring these epidemiological characters and also published a book called Disease in Plants, where he revealed about the knowledge of epidemics and various other kinds of things. This is Stockman physiological specialization, J.C. Walker phenolic theory, Muller and Berger coined the term phytoxin. Bengal famine around 1942-43, around 1.4 to 4 million people are dead just because of food starvation. J.E. Vanderplank, he is generally considered as a father of plant disease epidemiology and is also considered as a father of quantitative plant disease epidemiology. He was the one who gave the concept of vertical and horizontal resistance in 1968 and also coined the term vertifolia effect. It is generally means loss of horizontal resistance while breeding for vertical resistance is known as vertifolia effect. And he also introduced the many terminologies like simple interest diseases, compound interest diseases, monocyclic or polycyclic, that is called generally vertifolia effect, area under disease progress curve. All those things was explained by J.E. Vanderplank. He actually published 5 books. Among them, which the first one is very famous, Plant Disease Epidemics and Control 1963, and other books like Disease Resistance in Plants 68, Principles of Plant Infection 75, Genetics and Molecular Basis of Plant Pathogens 78, Host Pathogen Interaction 82, and also gave the concepts like protein for protein hypothesis, strong gene weak gene hypothesis, and disease triangle. These are all the various kinds of things that was given by J. Vanderplank. The first computer stimulation model is the epidem for early blight of potato and tomato on 1969, Diamond and Wagner and Horsfall, not Diamond and Wagner, probably Wagner and Horsfall, I think so. 1975, Krause et al. Developed the first forecasting model, that is a lead blight of potato. 1975, Nagarajan and Singh, they formulated Indian stem rust rules, whereas Nagarajan and Joshi identified the Puccinia pathway. And 1983, first expert system that was developed in USA to detect against 17 soybean diseases of 17 soybean disease in USA, that is called plant DS, I think so. Now we will discuss about applied plant pathology. Cato, this preferred the fumigation for the trees with bitumen and sulfur. Homer, they mentioned the therapeutic properties of sulfur. Remnant started the seed treating with sodium chloride. See, these are all the facts. They may ask what they have done, who have done, such kind of things. So please remember, Holmberg recommended the mercury chloride for the preservation of wood. Prevost copper sulfate against wheat burnt. Robertson sulfuric acid for the control of peach powdery mildew. Kendricks, proposed the lime sulfur preparation against grapes mildew, boiled sulfur for the control of powdery mildew. Garrison, Arnold, and as Edmund Thenard, they used carbon disulfide for the soil fumigation against phylloxera vastratrix. And Gaston, they designed the fumigation gun for this delivery or application of carbon disulfide. And PMA Meladat, we know, we might have studied this man, like he's the one who discovered this Bodomicher. So Bodomicher was discovered by PMA Meladat for the control of which disease? Grapes downy mildew. Grapes downy mildew is caused by Plasma paraviticola. So this is also serendipity discovery. Serendipity discovery means accidental discovery. So the composition of Bodomicher is around 1:1:100. 1 kilogram of copper sulfate, 1 kilogram of hydrated lime, and 100 liters of water all mixed together and can be applied on the crop. The only problem with Bodomicher is you cannot use more than 1 day. We must use it on or before 1 day. So the original formulation that was given by Meladat is around copper sulfate 5 lb, hydrated lime 5 lb, water 50 gallons. If you put all these things together, again it's same 1:1:100 only. So what is the chemistry behind copper sulfate plus calcium hydroxide, cupric oxide plus CaSO4? So this is generally. And Wazane, he is the one who tried copper sulfate in India at Pune for the management of various kinds of diseases like sorghum smut and all the things. And Mason, the person who discovered the burgundy mixture. Burgundy mixture was discovered by Mason. So burgundy mixture is also known as soda borax. This is the question that was asked in 2018. Burgundy mixture is also known as soda borax. So the composition of boda mixture is burgundy mixture is also 1:1:100. Instead of lime, we are replacing the sodium carbonate here. So in Boda mixture, so instead of sodium carbonate, lime will be there, which means copper sulfate, lime, water, Boda mixture. Copper sulfate, sodium carbonate, and 100 liters of water, that is the burgundy mixture. And Jensen developed the hot water treatment, and Trillett developed the fungicide proportions of formaldehyde. Sanders, Bowley, Selby, Lawrence, they have their own contributions here. And Beverly was the first person who identified or who developed a chestnut compound. So the composition is 2:1, 2 parts of copper sulfate and 11 parts of ammonium carbonate that can be used for damping of diseases. Narasimhan, they used a linseed oil like in bodomixture just to improve the coverage. Narasimhan in Indian who mixed the linolenic oil to improve the coverage and also increase the shelf life by 1 day. And Windling is suggested the trichoderma as a first biocontrol agent. Tisdale and Williams world's first patented fungicide is the theorem. So this theorem was developed by Tisdale and Williams in 1934. In 1942, Singh, they developed a Chaubetia paste. Chaubetia paste that was just developed by Singh in Almora district of Uttar Pradesh. And Chaubetia paste, generally the composition is around copper sulfate, copper carbonate 800 grams, red lead 800 grams, linolenic oil or linseed oil 1 liter. So Diamond and Horsfall, they discovered alkaline dithiocarbomates that is widely used as a fungicides. J.G. Horsfall, he developed a mechanism called fungicidal action, how the fungicide will work, or the mechanism of action of fungicides, and he wrote a book called Principles of Fungicidal Action. It's a very important book. Kittelson, it is very important. This was the repeatedly asked question. There is a person called Kittelson. He developed a fungicide called Captan. We can also call it as Kittelson killer. It is one of the most widely used fungicide for a seed treatment that was developed by Kittelson. That's why it is called as Kittelson killer, invented here in 1952. It is the first heterocyclic nitrogen compound. Rachel Carson, she wrote a book called Silent Spring. That was the movement of environmental movement. In fact, she had done lots of contribution to the environment because at the time when she was working as a working on this, she identified it is very carcinogenic to all aquatic and as well as humans and various other kinds of microbiota which is there on the earth. And she worked on this environmental movement. Anyway, she died in 1964 because of breast cancer, and later US identified the pollution effect that was caused by DDT, and later DDT was banned. That is secondary. And Van Schemling and Marshall Polka discovered the world's first systemic fungicide that is carboxin and oxycarboxin. Phosphatyl aluminum was developed in 1977, 1981 Fungicide Resistance Action Committee. So 1996 first strobilurin group of fungicide were launched. What is strobilurin group of fungicide? They are new generation fungicide which is actually isolated from a mushroom called Strobilurus tenacellus. This is a mushroom actually. And there are some acts. 1912 Plant Quarantine Act in USA. In India, the act was proposed on 1914 with the name of DIPA Act, Destructive Insect and Pest Act in India, very important one. 1946 DPPQS, Directorate of Plant Protection, Quarantine and Storage, located at Faridabad. 1966 Seed Act, 1968 Insecticide Act, 1968 Insecticide Seed Rules, 71 Insecticide Rules, 72 Federal Environmental Pesticide Act, 83 Seed Order, 96 Food Quality Product Act, 2001 Protection of Plant Varieties and Farmer Rights Act, 2002 National Seed Policy. Next one is biocontrol agents. Van Tubeff is the first person who introduced the term biocontrol agent. He is also considered as founding father of plant pathology in Europe. Van Tubeff first used the biocontrol, as I told, and Stanford suggested the control of potato scab by green manures. That was due to actually the biocontrol properties which was there inside. And Millardet and Taylor, they reported the potato scab, that is Streptomyces scab, can be managed by sterilizing soil and soil inoculation with one more thing, that is Streptomyces prasiox. And Stanford and Broadcast first used the term biocontrol agents and the suppressive effect in plant disease management. And he is a very important person. He is the first person who suggested the use of biocontrol agent, that is the trichoderma viride, against soil-borne pathogens like trichoderma lingorum. Some biocontrol agents like antagonists like trichoderma lingorum, against or trichoderma species against Rhizoctonia solani, Phytophthora parasitica, Sclerotian ralphsi, and suggested the use of trichoderma as a biocontrol agent. And they also control the Rhizoctonia, which causes various kinds of damping off and other kinds of root rots and all those things. And S.D. Garrett, he is largely considered the father of biocontrol agents when it comes to pathology, and is also considered the founding father of root pathology. And he gave the term competitive saprophytic ability, inoculation potential, and suppressive soil, various kinds of things. And he wrote books like Root Disease of Fungi 1944, Soil Fungi and Soil Fertility 63, Biology of Root Infecting Fungi 56, Pathogen Root Infecting Fungi 1970. And his work mainly worked on wheat take all disease that is caused by Gaumonomyces graminis tritici, and identified he told that by using a biocontrol agent there is only one disease we can control 100%, that is the wheat take all disease. The biocontrol agent is Pseudomonas fluorescens, and also studied the pathogen like Plasmodium brassica, Almillariella amelia, Armillariella amelia, Helicobacterium purpureum, Agaricus bisporus, Serpula lacrimus, various kinds of fungi have studied. And Bran and McGrath, they isolated the first antibiotic viridin, that is isolated from Trichoderma viridi. 1963 first commercial available biocontrol agent, that is a Penniphora gigantea. We can also call it as Plebiopsis gigans against a pathogen called Heterobacterium anosum. New and care, they introduced the first commercial available biocontrol agents against the crown gall disease by using another bacterium called Agrobacterium radiobacter. Agrobacterium tumefaciens is the disease causing, radiobacter is the biocontrol agent. Tumefaciens means tumor inducing, radiobacter is the biocontrol agent. Okay, Baker and Cook, they wrote a very famous book called Biocontrol of Plant Pathogen 1974. The developments and various kinds of teaching trainings, the pathological extension programs in India. In 1857, actually, the first teaching in plant pathology was started by University of Calcutta, Bombay, and Madras, and they started, working with the taxonomy and various kinds of things. In 1930, in the first time, the University of Allahabad, Lucknow, and Madras, they introduced plant pathology as a course. In the later 1945, Agra introduced the postgraduate studies, especially on plant pathology. Later in Government College, Kanpur. 1947, teachings of plant virology was started in ICAR at 1947. 1960, everyone understand the importance of teachings, especially related to mycology, bacteriology, and virology, so that they started working on both undergraduate and postgraduate programs to strengthen the agriculture sector in India by establishing the agricultural universities in India. In 1970s and 80s, they worked on specialized books that was published in plant pathogenic fungi, bacteria, various kinds of publications, and all types of diseases what actually responsible that is caused by the various kinds of pathogens in both field, horticulture, and vegetable crops. In 80s and 90s, introduced the computerized models, databases, computerized systems, expert system for the diagnosis and management of plant diseases. And some professional plant pathological societies and their publications now you see. And these are a couple of societies. In 1891, this is the oldest plant pathological society, a Netherlands Society of Plant Pathology. On the right side, we have given the journal name and bi-monthly or monthly or whatever it is. And first society is 1891, Netherlands Society of Plant Pathology. It's a very important one, and this is also one of the probable, and this is also the question that was asked in 2023, I think so. 1896, British Mycological Society. 1919, American Pathological Society. 1916, Pathological Society of Japan. 29, Canadian Pathological Society. 29, Chinese Society of Plant Pathology. 1947, Indian Pathological Society. 16, in Australian Pathological Society. 82, International Society of Molecular Plant Microbe Interaction. 84, Association of Plant Pathologists in India. 84, again Indian Virology Society. Again 84, Indian Society of Plant Pathologists. 92, Society of Plant Protection Studies. So for further guidance or further information, students can refer this book. For further guidance or clarification of doubts and all kinds of things, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.